All right, so part A, and again, we what tissue is made up of, or the definition of tissues, histology, and then we know that there's 4 different types of tissue. We discussed that. Looked at figure 4.1. Looked at how microscopy works with human tissue. Took a look at types of electron microscopes and what how we can tissues in those 2 ways. And then we took a look at epithelial tissue and really what its definition is and somewhat what its function is, right? And that there's 2 different forms of it. One that can cover things or line, for example, organs, and some that can form glands, right? And so that's really where the rest of the slide is composed of. We look at the background of some of the characteristics, but then we look at a lot of the covering and lining epithelial tissue, and then we look at what glands are made up of. Again, epithelial tissue is the answer, but there's more to that, right? Then we finally stop there, and I asked you to look at all 5 of these in more detail, and so we'll cover this right now and then move on, okay? So, so here are the 5 characteristics that all epithelial will have, tissue will have. Polarity, contacts, it's got connective tissue that supports it. It has the capability of being avascular, and it has the capability of regenerating, right? Or undergoing mitosis to repair or replace cells, right? Let's talk about this one first, polarity, right? Polarity basically means a pole, right? That definition basically means there's 2 sides, 2 different sides to something, right? Like a North Pole and a South Pole. Same thing when we talk about in chemistry, polar covalent bonds mean there's 2 charges, one on either end. And so when we use this definition, we're talking about the cell side that make tissues and what side is what. Those are the poles, right? And there's actually names for both sides. The first one is apical, and the next one is basal or base surface, right? It's be commonly known as base, right? That's what that word basically means. Okay. So let's look at the apical surface. The apical surface is the free side. This is the one that will be exposed to any surface or any cavity. And what I'm drawing here, let's say, is a row of cells. And this row of cells lines the esophagus, right? So if I am eating food, right, food goes down from my mouth, down the esophagus to my, let's say, stomach. We would see a row of cells microscopically, right? I mean, we'd have to take a microscope, take a section, and look at it in the microscope. Does that make sense to you? The little red dot is the nucleus, right? Okay. Now, the reason I mention this is because there's actually 2 sides, talking about this, 2 sides to those cells, right? The free side would be the side that's touching the food here in my rough drawing here. That would be the apical side. Does that make sense? So all of this side here, or where the food would be touching that side of the cell, would be the apical surface or apical side. Conversely, the opposite end is what you would consider the basal side or the base surface. That's the one that's actually attached, right? So these aren't floating around, right? They're actually attached to something, and the base side is attached to what we call the basal lamina, okay? It's a sheet of basically molecules that attaches the base side to the underlying cells, okay? So what do you think are the underlining cells that the basal lamina is attaching the cells to? Can anybody tell me that? When it says underlining cells, what do they mean by that? So like this side, for example, right? Let's say that side of the cells, they're attached to something. The basal lamina is like glue that attaches that side to other underlying cells. But what are those types of cells? Connective tissue. Okay. Now we're going to see this in a greater example later on when we look at the integumentary system or the skin system, which we'll look at hopefully today. If not, we'll start tomorrow. But that's connective tissue. So that's what the definition of, or the function of connective tissue is to connect then epithelial tissue, right? So if you can think of, for example, the skin, what I'm touching, what you can touch yourself on your whole body. This top layer is epithelial tissue, but the reason why I can't just pull my skin off, right, is because it's connected or adhered to by connective tissue. Does that make sense? This is why I can do this, right? You pull skin, but then it comes right in place because there's connective tissue holding the top layer of epithelial tissue. Okay, here's the apical surface. So I'll draw one small cell that is part of the epithelial tissue. So the apical surface is right up here, and the basal surface is right down here. And all of this down here is all connective tissue, adhering the top tissue or the epithelial tissue. When you start looking at slides today in lab, what you're going to look at is try to find where, if this is a epithelial tissue, you're trying to find this, because that's what you want to look at. But you always notice there's some cells underneath that are different, and that's, of course, the connective tissue. Okay? We know this already. This is another characteristic, right? Specialized contacts, but we know, for example, that the tissues need to fit closely together. So if there's two tissues just like this, they would actually look like this to you. But we know that there is connections, for example, in between here. And we call those junctions, right? Cell junctions. And we saw that in chapter 3, right? And the two cell junctions that we talk about there that hold those two cells together, one next to another, that make that sheet, are the tight junctions and the desmosomes. Remember, the tight junctions rope the plasma membranes around and hold them really close together. And the desmosomes kind of lock it in like the zipper, right? And we know that occurs there, and that's how we can make a whole sheet, let's say your skin. So we just discussed this, right? Supported by connective tissue, which means the underlining tissue below the epithelial tissue layer is supported by connective tissue, right? Below the basal lamina, we have a name that we talk about when we talk about that row or that collection of those cells, and we talk about those being the reticular lamina. Reticular lamina is a collection of connective tissue that has collagen in it. Collagen fibers. Collagen and collagen fibers are the same thing. And that supports the upper, right, epithelial tissue. Okay. So if I was looking at a side view of the tissue, this is, let's say, skin, just for our example. The top red squares are your epithelial tissue. Lying underneath is that basal lamina, which is kind of like a composition, like a glue. And underneath that is the reticular lamina, which is made up of collagen fibers. Okay? When you talk about both of these bottom blue areas together, that's called the basement membrane. Okay? The basement membrane. And the basement membrane is those two layers that hold up or support the upper epithelial tissue. Okay? Epithelial tissue is, in fact, avascular, but innervated. What does avascular mean? Without blood vessels, thanks, sir. Without blood vessels. Right? It's telling you that epithelial tissue does not have blood vessels directly feeding oxygen, nutrients, taking out waste from the epithelial tissue. They are nourished or getting that oxygen because they still need it, getting those nutrients because they still need them, transporting the waste out of themselves because they still do that by the connective tissue that's underneath it. And we do that through a process of diffusion. Okay, that's why we talk about diffusion, because this is another way we can see it, right? Diffusion, which is a high concentration, a low concentration. Sometimes we can transport that out of the cell, and that goes to the connective tissue where you find the capillary bed or those blood vessels that are going to pick up the waste or drop off the oxygen and nutrients. Epithelial tissue do have a nerve supply then. So they are fed by nerves. So they do sense things from the epithelial tissue. And finally, they have the capability of regenerating, right? So they have the capability of repairing themselves greatly or replacing themselves if needed. Because when you look at the place where you find epithelial tissue, many times that place where you find epithelial tissue is always exposed to some kind of friction, force, things like that to then cause either disruption or breakdown of that tissue. So it has the capability of replacing itself pretty quickly and easily. For example, lining our mouth and our throat is a layer of that epithelial tissue, and in fact, it can be rubbed off, damaged, and be regenerated, and that's a natural process because in the process of, let's say, eating food, drinking things, swallowing things, it's going to rub or have friction against the side of that wall. Some of those cells will fall off or get damaged, and they can regenerate. And that's a process that occurs naturally in the body. Got to be replaced, right? So that's what we talk about when we replace. And the way we replace cells is we grow them under cell division or mitosis, right? You need a lot of nutrients to do that, and obviously that's where the nutrients get, taken to them, and that's what you see. Okay? So let's talk a little bit about classification before we move on. But when we talk about classification, this is going to come into play this afternoon when you start looking at tissues, because there's going to be a lot of tissues to look at of all of the 4 types of tissue. And so when we look at this, we have some rules involved as far as how we name them, how we classify them, right? The first name of epithelial tissue is always going to be indicating the number of layers that the tissue will have. If it is simple, that means there's only one layer. Very easy to remember, right? Simple means just one layer of cells. The examples I drew, again, various sizes there, but they're all one row or one layer. Okay? Now, if you're stratified, it's 2 or more layers. So let's say on top and bottom, we've got another layer down here. And that would be a stratified layer. There's 2 layers of epithelial tissue. Or more, obviously. There's 4 or 5, and you'll see that. For example, in skin, you'll see there's upwards of 30 layers, right? Of the very top layer of epithelial tissue. So that's the first name. Then the second name indicates the shape of the cell. Okay, what it looks like as far as shape. And there's 3 main shapes, and there's actually more out there, but as far as we're concerned, the majority of our cells fall under these 3, right? A squamous cell is flattened, scale-like, looks like a pancake. It's how I've always remembered it, okay? Really flat cell, okay? Still has all the parts that a cell should have. That's not changed, but the shape is real flat. Cuboidal, box-like, cube shape, and columnar, column-like, or pillar-like. Okay. So here's an example of a simple versus a stratified. This would be, for example, you would see this, for example, when we look at the respiratory system and look at the lung tissue, the very edge of what we call the alveoli, which are the smallest units that would transport oxygen inside blood and then carbon dioxide out. The layer there of epithelial tissue would look like that, flat cells making the wall. And then this is, of course, would be some skin layer, right? Would be our layer of the skin, would have many of these layers. And you can see that some of them look like cuboidal there. And the farther you go, some of them look flat, squamous. And that's actually how skin works. And we'll get into that in chapter 5 because we'll see how those layers work. And you can see, obviously, on both of them, there's the basal surface and there is the apical surface. The free side versus the adhered side there. Figure 4.3b shows you the different shapes, right? There's a flat, there's the cube shape, and there's the column shape, right? Okay. So let's break into groups now and discuss what we were talking about all the way to this point. Let's do 4 minutes, right? Do 4 minutes. Okay, we can start now, okay? So you can wake up here, right? All right, so at this point, it starts talking about each of the epithelial tissue. And so we'll just look at the first one so I can show you what you need to study from, right? Or what you need to look at, right? So this talks about simple squamous, right? That's our very first one. Okay, so if you study the shape and the arrangement of the cells, then you know that this is flat cell, it's one layer, right? So that's really what that is, right? Number one, that's what you'll need to make sure to understand, right? Which, again, if you look at the rules for epithelial tissue, you should already know that, right? the function, right? This one is made for places where there's going to be a lot of diffusion, where things need to go from one area to another area fast, like molecules, for example. Bless you. Kidney and lungs, where you find them, right, as an example. And that's kind of what you see in this figure, right? You look at the photomicrograph, which is great and all. That's what we're going to look at in lab. This is what you're going to see over and over and over again, both in lab, in the microscope, and in your textbook, right? But then this talks about basically the shape, size. There's the function, right? Where you find it, right? Okay. Same thing will apply for each one of them, right? So as you go along, here's simple cuboidal. You know what simple cuboidal is? Single layer cube shaped cells, because that's what these two words basically mean, right? Here's the function where you find it, right? That makes sense, right? Now, if you know what the kidney does, right? Kidney filters, secrete stuff, right, through filtration, which means we take bad waste that we don't need and we stick it into the on the other side of the kidneys, and that becomes urine. Does that make sense? The stuff that we talk about waste travels through these cube-like cells here. So this would be waste that goes from bloodstream into the kidneys and then goes through these, and that becomes urine. Does that make sense? So lining what we call the tubules, which is the kidneys, basically a little tube that does this, are going to be a bunch of these simple cuboidal cells. That's all they do. That's what you need to know as far as function, right? That's what about there. The ones right here, let me just draw one here. But you see there's about, in that circle, 4, right? The reason I know there's 4 is because you see the purple dot, you know that's the nuclei, right? You can't see the line. I mean, if you really depends on where you see it, but let's look at the one next over. You can see where one cell starts and one cell ends. Can everybody see that? Okay. Those are simple cuboidal cells. Now they make a circle like this, which is not, I mean, that doesn't, what we just talked about didn't describe if it makes a circle or if it makes just a nice little tube or whatever. But nonetheless, there's a single layer of cube cells. And that's an example. What we actually did is actually take a section of kidney just like that, and that's what you're looking at under the microscope. That's the tube. And we're looking at, let's say this is the tube and I cut it, and that's what you're looking at. Does that make sense? Surrounding the tube, simple cube. Okay? On and on. So it goes over a couple of them. And really what you know from these epithelial cells. Okay? So again, I'm not going to go through that. It's just that's what you need to pull out from that information in your reading when you look at the book and when you look at the PowerPoint. Okay? What I do want to move onto is glands, right? So what I breeze through, or what you're going to look at, is all the covering and lining epithelial tissue, right? Because there's two forms of epithelial, right? Tissue. The covering and lining that we just went over really quick, and the ones that form glands, right? Now these cells, or these collections of epithelial cells, make a gland and they secrete what we call a secretion. This is the fluid, which is aqueous based. Does anybody know what that means? This really pretty word, aqueous. Water. It's a water-based fluid, okay? Called a secretion. Now, depending on what gland this is, that secretion can be, I don't know, breast milk, right? Can be sweats, can be oil. Does that make sense? All three of those are secretions. All of those are aqueous. They're water-based. And all of those are three examples that the human body can make and then secrete through these cells. Lining this area is what we call those glandular epithelial, and they're the ones that are responsible for making this secretion. Okay? Now, we can classify it as far as where we secrete it. And this talks about if you're an endocrine or an exocrine. And the reason I stress that exo versus endo means that describes where the site is. Endo means inside, internal secretions, and exo means external secretions. Okay? If you're an endocrine gland, and you're the cells that line an endocrine gland, you're secreting inside. Here's a great example of that. Hormones, for example, secrete inside, and exocrine glands secrete outside, and sweat is a great example of that. Now you can also name your gland depending on the number of cells forming the gland. If you're a unicellular gland, that means you're just one cell making one product, secreting one product out. If you're multicellular, you're a collection of many cells that all have the same function. They all secrete whatever they secrete out of that gland. Now I just want to show you how a gland will form, basically, from epithelial tissue and in the process of what we call invagination, or folding in, if you will. Now you can see the layer of epithelial tissue here, and you can see it invaginating or folding in like this. And eventually it'll bulge out and you'll have this exocrine gland. Can everybody see that the row cells are still the same row, but they're specialized because they're going to make something. And let's say this is a sweat gland. And this top area right here would be the sweat pore. And this top area right here would be your surface of your skin. Does that make sense to you, right? And those cells are actually making, the black arrows, let's say, are making sweat itself. The cells themselves that are lining the gland are making the sweat. They'll add to this area here, okay? And because of a little smooth muscle or stimulation for the nervous system, they push the sweat out. Make sense, right? Get that part so far? Now, sometimes they'll form this invagination and they'll cut off, right, in development, and then there'll be a collection of cells underneath, interior of the body, and those cells are responsible for collected there and responsible for secreting, making and secreting a certain type of chemical. And that chemical would be let's say a hormone. And notice what's on top or surrounding those collections of cells. The bloodstream, because those cells will secrete their hormone and will catch a ride in the bloodstream to be used somewhere else. Other cells will use them as stimulation to make or do something for the body. But just to look at the structure is completely different, right? Although it does look the same. And this is in development, right? This is forming endocrine gland cells. We call these ductless glands the endocrine glands because they don't have a duct to them. They're released in the interstitial fluid and then they're picked up by the bloodstream. And so we can define hormones right here, although we talk more about it in AMP2, right? But hormones are chemical messengers, right? They can travel not only in lymph, but in blood, right? And we use this as a signal to other cells in the body to stop what they're doing or start what they're doing. And that's what hormones really do. Exocrine glands are usually, their secretions are released on body surfaces, skin, body cavities sometimes. We have many more of these than we do endocrine glands. So we have more exocrine than we do endocrine. They secrete the product into ducts, and then those ducts can push out the secretion. So not only sweat and oil, but a great example of that is a mucus gland, secretes mucus, or salivary gland that you find in the mouth that secretes then saliva, right? So those can be one cell by itself or many cells that make up the gland. The two big examples that we have in the body for unicellular glands are the mucus cell and the goblet cell. The mucus cell you find in the intestinal wall, and the goblet cell you find in the respiratory tract. Both secrete mucin, okay? It's a sugar protein motif, which means it's a molecule made up of a sugar and a protein put together. And then we add water to it, but it makes mucin, and then we have water there, and once the mucin hits the water, it becomes mucus, okay? So what is mucus made of? Mucin and water. The mucin makes it sticky. Does that make sense? Now, if I have mucus, right, and we know that mucus has different consistencies, right? You know this in life. You know that sometimes mucus is real thick, sometimes it's super thin. So what's the difference between a thick mucus and a thin mucus? What's the difference there?
The thicker one's more sticky, which probably means it has less water.
Good. And the other one has what?
More water.
More water. That's what that means. Does that make sense to you? The water composition is going to be different because it's different. We have a different characteristic, more sticky, thicker, right? Or thinner, not as sticky, more watery. Okay. Here's your typical unicellular cell. This is a one goblet cell, by the way. And you can actually see it here. This is one goblet cell. That is one unicellular. And that's the drawing. Actually, it looks almost exactly like the drawing, which is really cool. Look at huge amount of nuclei. It's a big nuclei. What else is big in as far as not only size but number? What do you see most of here in the mucus cell? If I was to look at the drawing here, what do you see most numerous here inside the cell? No, not lysosomes. The Golgi apparatus. Here's the Golgi body, right? So what is happening is that the rough ER, or the nucleus, is sending instructions to the rough ER to then make a bunch of that sugar protein stuff, gets sent to the Golgi apparatus, and the Golgi apparatus is modifying it, right? And like That's what it's supposed to do. And then pinching it off, right? And making what we call vesicles. Those are those little circles. And inside those circles are pretty much all mucin. Does that make sense? You see how it's holding there for right now? And if we stimulate or something comes by or whatever happens, we stimulate in that area, these are going to all release out. Combine with water and form mucus. Does that make sense? That's just to show you chapter 3, the cells and why vesicle, and we didn't even get into exocytosis and endocytosis, but that's exocytosis. Vesicles being brought out, releasing what they have inside, which is all mucin, basically. All that green little dots have mucin in it, and releasing and releasing and releasing. Okay. Now, multicellular exocrine glands means there are many cells that make up this gland. They're going to have a duct. They're usually supported by connective tissue, and there's going to be a blood supply to them usually, and a nerve supply to them. They form a lot of different ones. I'm going to show you this example. I don't expect you to study these. It's just to look at them to say, oh man, there's all different types of shapes and sizes. So don't study this, okay? Just put a big X, but don't study it, right? You don't need to know, you don't need to memorize any of these names or where you find them, so on and so forth. But just to kind of show you that that's a salivary gland right there. Almost looks exactly like a mammary gland. That's where breast milk is made, for example, in the breast. Different than, let's say, an oil gland. There's a stomach gland there. It produces stomach secretions that line the stomach. Those help break down food. Those cells that are kind of reddish are different cells that secrete different products or enzymes that break down food. Same thing with these in the small intestine. That's what lines your small intestine, these glands, and they have secretions that, when released, will start to digest food or break down food there. Okay? All right.